Hello students, I welcome you all in today's session. In today's session, I will be teaching you biology. Name of the chapter is biological classification. In today's session, we will be discussing about the need of the classification system, various types of classification system. Then we will be discussing about the kingdom Monera. Okay, so now let's get started. So learning objectives I have already told you. In this chapter, we will be discussing about 2 kingdom classification, then 5 kingdom, what were the bases of the 5 kingdom classification, and we will be also discussing about kingdom Monera. So let's get started. So I hope all of you are already familiar with the term classification. We use it in our daily life also. If you see your kitchen, there you might be observing that your mother has very meticulously arranged the different type of utensils. Spoons have been kept separate, bowls have been kept separate, plates have been kept separate, and if you want to find a particular type of utensil, it's very easy for you. So similarly, we also do the classification of living organisms also, that is very important. So the first classification system which came up, that was given by Aristotle. I hope you might have heard his name. He was a very famous Greek philosopher who is also known as the father of biology. So he was the first one who had done the classification, and how he had done, he had divided the plants into 3 categories: herbs, shrubs and tree. Okay, and he had divided the animals on the basis of color of their blood, whether they are having red blood or they are having without blood. So if you are observing a plant, just by observing the plant you can tell whether the plant is a herb or a shrub or a tree, depending upon its size, depending upon the thickness of the stem. And animals also, it's very easy, we can easily tell whether they are having red blood or not. But this classification system, as and when the time progressed, was not found to be adequate. And then we came up with another system which was given by Linnaeus, and this system was given in 1758. Okay. Now Linnaeus' name also you have already heard that he had given binomial classification, binomial nomenclature, and this Linnaeus gave a 2 kingdom classification system in which he divided all the living organisms into 2 kingdoms. The first kingdom which he gave was the kingdom Plantae, in which all the plants were kept. The second kingdom which he gave was the kingdom Animalia, in which all the animals were put together. So talking about the advantages of this type of classification system, this is very easy. Even a layman person can classify a particular organism into plant or animal. But as and when the time progressed, this system was found to be inadequate, because it has its own set of limitation. Like if we are talking about unicellular and multicellular organism, this system is not able to differentiate between the two. And in class 10th, you have already learned the definition as well as examples of unicellular as well as multicellular organism. Similarly, this system cannot differentiate between prokaryotes and eukaryotes, right? Prokaryotes, eukaryotes term you have studied in class 10th also, and it is very unjustified. It is totally unjustified to put the prokaryotes and eukaryotes together, but this system had clubbed them together. So that is why it was found to be inadequate. Then similarly, if we are putting the green plants which are showing photosynthesis along with fungi, fungi you know that they do not contain green pigment chlorophyll, so they have a different mode of nutrition, so we are clubbing them together. Do you think it is justified? No. So that is why a demand started arising, and this system got replaced by 5 kingdom classification, which was given by an American ecologist. Name of that person is R.H. Whittaker, and he gave this classification system in the year 1968. Okay. Then we also have 3 domains of life, which was given by Carl Woese, that we will be taking up later. So students, we were discussing the shortcomings of the 2 kingdom classification. So in the 2 kingdom classification, different type of organisms have been clubbed together. As you can see in point number 1, bacteria and blue-green algae. Actually, blue-green algae is a cyanobacteria. It is a type of bacteria only. So do you think it is justified that we place prokaryotes, because we know bacteria are prokaryotes, and we are clubbing them with other organisms which are eukaryote. So clubbing of prokaryotic and eukaryotic organism is not justified. Similarly, you can see that here it is fungi. This whole group was placed under kingdom Plantae just because they are having a cell wall. But we know fungi is very different from the plant. It does not contain the pigment chlorophyll, and its mode of nutrition is also different. So it is not justified to club fungi along with the plants. So what R.H. Whittaker did, he separated this bacteria and placed them into a separate kingdom. That kingdom was known as kingdom Monera. Similarly, fungi, they were placed in a separate kingdom, and the remaining unicellular eukaryotic organisms, they were placed in another kingdom. That kingdom was known as kingdom Protista. So that is how this 5-kingdom classification system came into being. Then similarly, we have also discussed that it is not justified to place the unicellular and multicellular organism together, and this also we have discussed. So now shifting to the next portion. So criteria, now let's discuss what all criteria R.H. Whittaker had used for his 5-kingdom classification. For his 5-kingdom classification, he has used these main points. The first one was the cell structure. Okay, cell structure means whether the cell is eukaryotic or prokaryotic. Then what type of thallus it is having, what type of body organization is having, mode of nutrition, whether it is autotrophic or heterotrophic, then what type of mode of reproduction it is having, phylogenetic relationship. Phylogenetic relationship means we were trying to study the evolutionary relationship between the different groups. So this, by using all these criteria, R.H. Whittaker had proposed a 5-kingdom classification system. So this table is very important. If we will discuss this table, with the help of this table, you will be able to understand how we have classified the living organisms into 5 kingdoms. So the first point which we have is, that is the cell type. Okay, I hope all of you remember these terms prokaryotic and eukaryotic. You have studied these terms in class 10th as well. So prokaryotic means having ancient type of nucleus, means these organisms, they do not have a well-defined nucleus. Nucleus is not covered by nuclear membrane. Whereas on the other hand, all the remaining 4 groups, they are eukaryotic. Even fungi is also eukaryotic. Even plantae is also eukaryotic. Then Animalia is also eukaryotic. So for remembering this, you can simply remember this thumb rule. Okay, imagine that this hand of yours is representing the 5 kingdom. The first kingdom is the kingdom Monera, which is having exclusively prokaryotic member. Remaining 4 kingdoms, we have Protista, we have fungi, we have plantae, we have animalia. All the 4 kingdoms are eukaryotic. Okay, so first point should be clear to you that out of the 5 kingdom, the first one is prokaryotic, the remaining 3 are exclusively eukaryotic. Now this third point, we will be discussing it along with this only, because these 2 points are related. Obviously, any organism which is prokaryotic, it will not be having a well-defined nuclear membrane. So here we have written nuclear membrane is absent. And as I have already told you, the 4 remaining kingdoms, they are having eukaryotic members, so nuclear membrane will be present. So you can club the first point and the third point, and you can study them together. Then we have next point, that is called cell wall. I hope you remember in class 10th you people had studied the difference between plant cell and animal cell. There you had learnt a very important difference, that plant cells contain cellulosic cell wall, whereas in case of animal, cell wall is absent. Okay, but it is not necessary that every organism will be having a cell wall which is made up of cellulose. Like for example, if we are talking about Monera or bacteria, they have a cell wall which is made up of peptidoglycan. Now let's see what does this word peptidoglycan means. Peptido word refers to protein, glycan word refers to polysaccharide. So this type of cell wall which is made up of proteins and polysaccharide, this is found in bacteria, and this is called peptidoglycan. Then when we are talking about Protista, not all Protista have cell wall. Some of them are having cell wall, but that cell wall is present in a slightly different form. Just some of the protists, they have a covering, and this covering is called pellicle, and this pellicle is actually made up of proteins. So we are finding that the covering is having a different composition. Then let's talk about next kingdom, that is the kingdom fungi. So in kingdom fungi, the cell wall is made up of another compound, and that compound is known as chitin. Okay, so we now you will realize that it's not necessary that the cell wall of every organism is made up of cellulose. Either it could be made up of peptidoglycan, or it could be made up of pellicle, or it could be made up of chitin. Okay, now let's talk about point number 4, that is the body organization. Talking about body organization, you have already studied in your lower classes that all the organisms, they are made up of cells. Cells are arranged into tissues. When tissues will join together, they will form organs. When various organs will join together, they will form organ system, right? So now we have to discuss about the level of body organization in all the 5 kingdoms. In the first kingdom, because it is a unicellular organism, so body will be having the simplest level of organization, that is cellular, right? Then talking about protista, since they are also made up of single cell, so body will be having cellular level of organization. Then talking about fungi, because they are multicellular, but here you can see that all the cells, they have joined together to form tissue, so they are having tissue level of organization. Then plants may either tissue or organ level of organization may be present. Animals may we can have all the 3 types of organization, tissue also, organ system also. So this these 2 terms, these 2 kingdom you will be studying in detail in chapter number 3 and 4 also. Now coming to the last point, which is very important, that is the mode of nutrition, how they are obtaining their food. So on the basis of mode of their nutrition, we can divide them into 2 broad categories. Either they could be autotroph or they could be heterotroph, okay? Autotroph may also, they have mentioned 2 subcategories, that is chemosynthetic and photosynthetic. This I will be taking up in detail when we will start with kingdom Monera. Heterotroph may either it could be a saprophyte or parasite. So I hope you remember autotrophs are the one which can prepare their food on their own. Heterotrophs are the one which cannot prepare their food on their own, they are dependent on others. Then talking about protista, either they could have autotrophic mode or they could have heterotrophic mode, but fungi may no autotrophic mode will be present, they are exclusively heterotrophic because they do not contain the pigment chlorophyll, so either they will be having saprophytic mode or they will be having parasitic mode. Plants may, majority of the plants are autotroph, leaving a few exceptions. Most of them can prepare their food on their own. Then coming to the kingdom Animalia, they are heterotrophs, means they cannot prepare their food on their own, they are dependent on other organisms. So students, now we are starting with the first kingdom, that is the kingdom Monera, okay? So kingdom Monera may, as we have already discussed, it contains unicellular prokaryotic organisms, and in this kingdom bacteria are the sole members. Only bacteria are present in this kingdom, because we know bacteria are unicellular as well as prokaryotic, and we can divide this kingdom Monera into 3 classes, into 3 groups. The first group which we have, that is your archaebacteria. We will be discussing them in detail. Then second group which we have, that is eubacteria. The third one that we have, that is the mycoplasma, okay? So which kingdom we have started with? We have started with the kingdom Monera, and in this kingdom bacteria, which are unicellular prokaryotes, they are the exclusive members, okay? Then we have types of bacteria. We can divide bacteria according to their shape. On the basis of their shape, we can have 4 different types of bacteria. Let's take first one, that is the bacillus. Bacillus word means rod shape. If any bacteria is having a rod shape structure, that type of bacteria would be known as bacillus. I hope all of you remember the name of the bacteria which is found in the curd, and at the end of the name of that bacteria the word bacillus is being used. So it is understood that that particular bacteria will be having rod shaped structure, and name of that bacteria is lactobacillus. So I think it will be very easy for you to learn the example of this particular shape, because this particular type of bacteria, because the example you are already familiar with. Now sometime it may be having this spoon like structure also. This is a rod shaped bacteria only, but if it forms a spore inside it, due to that spore formation it will have this appearance of spoon like structure. Then second group which we have, that is the coccus group, okay? Coccus word means spherical. So any bacteria which is having spherical shape, that will be placed in this category. And coccus may we have the classical example of bacteria which is called diplococcus, okay? Di word meaning all of you are familiar with, that is 2. So if any bacteria is having 2 cocci arranged together, that will be called diplococcus. So example of this group will be diplococcus. Then we have third, that is the spirulum. All of you have seen spiral binding notebooks. So if any bacteria is having the spiral shape, that group will be called as spirulum. And in this case, flagella may be present at one, or it may be present at both the ends, okay? For this category, we have the example spirochete. This is a bacteria which is spiral in shape. So the example we can learn, that is the spirochete. Then we have the last last group, that is the vibrio, okay? Vibrio word stands for having comma shape. All of you have used comma in English language. So any bacteria which is having comma shape, that would be called Vibrio. And iska jo classical example hai, that is Vibrio cholerae. This is a bacteria which causes cholera. So this bacteria will be having comma shape, right? So what are the 4 groups of bacteria on the basis of their shape? The first one that we have is Bacillus. The second one which we have is Coccus. Third one which we are having is Spirulum. Fourth one is the Vibrio. An example of Vibrio is the Vibrio cholerae. This is a bacteria which causes cholera. So we can classify the bacteria on the basis of their shapes. Okay. Then we have to discuss about Archaebacteria. Actually, जो first group है, we have already discussed that bacteria can be classified into 3 groups. The first group that we have to discuss, that is the Archaebacteria. This Archaebacteria is divided into 3 further subcategories, and this, let's first understand the meaning of the term archae. Whenever we are listening any new term, we should get familiarized with its meaning so that it's easier for us to understand. Archae word stands for ancient. Okay. So any bacteria which is ancient can be placed in this category. Now they are also called the oldest living fossil. Now a question comes ki why we are calling them as oldest living fossil? Because these bacteria, they were formed when the earth was newly formed, and because they are living in such harsh conditions which were present on the primitive earth, and they are still living in such harsh conditions, so that is why these bacteria are called Archaebacteria, ancient bacteria. And we can divide them into 3 subgroups. So the first one which we have is halophile. Okay. Now let's understand the meaning of this word halophile. Halo word stands for salt or halogen. Phile word stands for affinity. So any bacteria which is having affinity for salt or which is a salt lover can be placed in the group of halophiles. So where we will find these halophiles? We will find them in the Dead Sea, where no fishes or any other living organism can survive, but these bacteria can flourish very well. Okay. An example of this we are having is Halobacterium. Then we have second type of Archaebacteria only, which is called thermoacidophiles. Okay. Thermo word refers to temperature, high heat. Acido word, I hope all of you are familiar that pH can be either acidic or basic or neutral. So acid word here refers to the acidic medium. So any habitat which is having high temperature as well as acidic medium, that and in that habitat, if any bacteria is surviving, that bacteria will be called thermoacidophile. Thermoacidophile conditions are found in the hot springs. So all those bacteria which are present in the hot spring water, they will be thermoacidophile, and the classical example for this is sulfobolus. Okay. Now let's shift to the next one. Then we have the third type of Archaebacteria, that is your methanogens. Okay. Methanogens word refers to methane generating. So all those bacteria which can generate methane, they will be called methanogens. Now these bacteria, they are anaerobes. Anaerobes means they do not require oxygen, and these bacteria, they are found in the swampy area. Wherever we will be having a marshy place, there these bacteria will be found and they will be generating methane. In fact, these bacteria are also found in the digestive system of grass eating animals. You might have heard the term ruminants. Ruminants are grass eating animals. So in the stomach of, in the digestive system of these grass eating animals, these bacteria are found, and there these bacteria are helping us a lot, because with the help of these bacteria, if we are storing their waste material in a biogas plant, we can generate biogas also. So they are used in the production of biogas also, and the example for this which we have is Methanococcus. So how many 3 groups of Archaebacteria we are having? The first one was halophiles, the second one was thermoacidophiles, the third one which we are having is the methanogens. Now a question must come in your mind that all these Archaebacteria, they are living in very harsh situation. It's not possible for any living organism to survive in such harsh situations. So how are they managing? So the answer of the question is that they have a different type of cell wall. Okay. In the beginning of the classification, 5 kingdom classification, I had told you that bacteria, they have peptidoglycan cell wall, but in case of Archaebacteria, the cell wall composition is slightly different. They have pseudomurine. Peptoglycan is also known as murine, but here the cell wall structure is slightly different. It is pseudomurine, and that is why they were able to survive in harsh situation. So with this we have completed the topic Archaebacteria. Then we have next main group of bacteria, which is called eubacteria. Now splitting the meaning of the word, EU word stands for true. So any bacteria which is true will be called eubacteria. Now what does this word true means? If we are talking about the eubacteria, they are having the typical cell wall which is made up of peptidoglycan. I hope you remember this meaning. Peptido word refers to protein, glycan refers to polysaccharide, so both of them are mixing together to form the cell wall. If they are motile, they will be having a flagellum also. Eubacteria, we also have cyanobacteria. Cyanobacteria word refers to blue-green algae. Okay. If you will ever see a water body, on the surface of the water body, sometimes it is entirely covered with the blue-green algae, and these are nothing, they are cyanobacteria. But the common name, blue-green algae, sometimes students, they tend to get confused, because algae word, if it is being used, they will think it is a plant. But actually they have been given this common name because they have pigments, chlorophyll A. This is a pigment which is similar to the pigments of higher plant, and by using this type of chlorophyll they are able to prepare their own food. Okay. So cyanobacteria mein, now we have to discuss the structure of a typical cyanobacteria, that is the nostoc. So this diagram we will be discussing. This is a algae which is living in colonies, means large number of cells have aggregated together. It is found in water. So whenever we are talking about this nostoc, the first part that we have to discuss, that is the mucilaginous sheath. If you will observe the diagram carefully, then you will be able to see that it is covered by a gelatinous or mucilaginous sheath which is present on the outside. And this sheath is very important because it is keeping all the cells of the colony together, and it is also providing it protection. Then if we are talking about these cells, these are the normal cells, and you can see that they are containing pigments inside them, and that pigment is chlorophyll A, and with the help of that pigment they are able to prepare food by the process of photosynthesis. But now students, please pay attention on this cell, and what is the name we have written here? Heterocyst. So these heterocysts are special type of cells. If we are comparing their size with the normal cell, size is comparatively bigger, cell wall is thicker, and these cells are playing a very important role because they help in the process of nitrogen fixation. I hope all of you have heard this term nitrogen fixation. This is a process in which the heterocyst cell or any bacteria is able to convert the atmospheric nitrogen into simple soluble form. So this heterocyst cell has all the enzymes, all the conditions which are necessary for the process of nitrogen fixation. Cyanobacteria are usually found in aquatic. So if they are growing in a paddy field, it will be very good for the farmer because they will be enriching the soil. They will be increasing the fertility of the soil because they can help in the process of nitrogen fixation. So now we have to discuss about the classification of bacteria on the basis of their mode of nutrition. It was there in the beginning of the 5 kingdom table also. There we had not done in detail. Now we will be studying it in detail. So bacteria can be autotrophic. Autotrophic, auto word means self, trophic means food, which can prepare their food on their own. And this autotrophic can be further subdivided into 2 categories. The first one that we will be having is the photosynthetic. Okay. So we have divided them into 2 categories on the basis of the source of energy. What is the source of energy they are using while preparing food? Okay. If they are using solar energy, then we will be placing them in the first category because photon word refers to energy of the sun. It is referring to the energy. So it is referring to light. So if they are using the solar energy, if they are using the sun energy, and by using that energy they are preparing their food, then they will be called photosynthetic. Then we can also have another category which is called chemosynthetic. Okay. Here we have used the word chemo. Chemo means they are using certain inorganic chemicals, they are oxidizing them, and whatever energy is being released in that process, by using that energy they will be preparing their own food. Like in class 11th, you have a chapter of mineral nutrition. You will be studying about certain bacteria which are playing a role in nitrogen cycle. There you will be finding that they are oxidizing nitrates, nitrites and other ions, and by using that energy they are preparing their food. So we can place them in the category of chemosynthetic. Second group which we have, that is heterotrophic bacteria. Heterotrophic means we can further subdivide these heterotrophs into two categories, parasites and saprophytes. Saprophyte means which are obtaining their food from dead and decaying organic matter. So if any saprophytic bacteria is present in the soil, it would be converting that dead and decaying organic matter into humus. Parasite means which are obtaining their nutrition from another living organism. So all those bacteria which are capable of causing disease, they will be placed in this category of parasitic bacteria. And for learning the name of different parasitic bacteria, I can give you a short trick, and that is CTCT. Okay. Now let's see what does this word means. First alphabet C refers to citrus canker. Okay, this is a bacteria which is a parasite on plants. Then we have typhoid. Again, this is a bacteria which causes disease. Then we have cholera. This is also a disease causing bacteria. So obviously it will be having parasitic mode. Then we have tetanus. So with the help of this trick CTCT, you will be able to learn the examples of parasitic bacteria. Now we have already discussed this term. Autotrophic bacteria we have already done, and we have also discussed that cyanobacteria, they are having autotrophic mode of nutrition because they have pigments similar to the pigments of plants. They have chlorophyll A, and thus they can prepare their food by the process of photosynthesis. In the last portion, students, now we have to discuss about the methods of reproduction in bacteria. The first method that we have to discuss, that is the fission method. Binary fission you all have studied in class 10th. Binary word refers to two. Fission means splitting, where the bacterial cell will divide and form two daughter cells. That process will be known as binary fission, and this is a very simple method of asexual reproduction. Then sometime bacteria, if it is present in unfavorable situation, if the situation is very harsh, if the situation is very unfavorable, then it may form spores also. Like we have seen in case of bacillus, that the bacteria was rod shaped, but because it has formed a spore, so it was appearing like a spoon-like structure. So if the conditions are unfavorable, then the bacteria may form a spore also, internal spore, and these spores can survive easily in the harsh situation. Once the situation resume back to the normal, these spores will be able to form bacteria. Then we have third method also, that is your sexual type of reproduction. Sexual type of reproduction mein what would happen, the two bacteria, they would be getting attached with the help of a tube-like structure, and with the help of that structure they would be exchanging a part of their DNA. So this type of primitive sexual reproduction, this is not a classical sexual reproduction because no gamete formation or no other procedure is taking place. They are simply exchanging a part of their DNA with the help of a tube-like structure. So this type of primitive sexual reproduction is also seen in bacteria. So now let's recapitulate what are the different methods. The first one which we have is binary fission. You can see in the diagram also. Then under unfavorable situation, it may form spore, and it may also exchange a part of its DNA with the help of primitive sexual reproduction. This process does not match typical sexual reproduction process because no gamete formation is taking place. So students, we have already discussed two groups of bacteria, eubacteria, archaebacteria. Now we are discussing the last group, that is the mycoplasma. Now these mycoplasma are very special because just like a typical bacteria, they do not have a cell wall. Cell wall is absent, and because the cell wall is absent, they can change their shape very easily, and that is why they are called jokers of the kingdom. Then second feature, which they are the smallest living cells. If we are comparing the size of mycoplasma with a bacteria, their size is even smaller than that of a bacteria. Then third point is that they can survive even without oxygen. They do not need oxygen for their survival. Then they are mostly capable of causing diseases in plants and animals. It means they are pathogenic. And the classical example for mycoplasma which we have is that is PPLO, pleuropneumonia-like organism. So in today's session, let's quickly recapitulate what all points we have covered. First, we covered the two-system classification which was given by Aristotle. Then we discussed two-kingdom classification which was given by Linnaeus. Then we discussed the five-kingdom classification which was given by R.H. Whitaker. And then we also discussed about kingdom Monera. In kingdom Monera, we had classified the bacteria into 4 groups on the basis of their shape. And we had also discussed their 3 main groups. First one was the archaebacteria, then we had eubacteria, then we had mycoplasma. So I hope you enjoyed the session and you also understood all the concepts. Thank you so much.